it is my absolute pleasure to introduce our guest speaker this evening, Dr. Peter Turnbaugh. Peter is a professor in the Department of Microbiology and Immunology here at UCSF, and he leads an interdisciplinary group of microbiome researchers that are fully committed to understanding host-associated microbes, reducing complex human microbiome ecology to molecular mechanisms, and applying the findings of his research to improve the practice of medicine. Currently, his group is very much engaged on focusing on the microbiome's role in pharmacology, in nutrition, and in phage biology. Peter received his graduate degree, his Ph.D. at the Washington University, St. Louis, where he studied under the incredible Dr. Jeff Gordon, one of the most preeminent researchers in the field of human microbiome research. But since his graduation, Peter has been equally impressive. He's received numerous awards and been named as the Damon Runyon Ratcliffe Cancer Innovator, Searle Scholar, and a Chan Zuckerberg BioHub Investigator, just to name just a few of the accolades that have been provided to him. And I'm super excited to hear this. I know this will be a great lecture. And tonight, Peter will talk about they are what you eat, how food and drugs interact with the gut microbiome. Peter, take it away.
Thanks so much, Sue. It's really a pleasure to be here with all of you virtually. Hopefully next time we can do this in person. And I'm going to share with you some of the sort of snapshots of the work that we and others have been doing, looking at the intersection between the gut microbiome, nutrition, and pharmacology. And then we'll sort of take a deep dive into one of our unpublished studies, looking at the role of the microbiome in cancer therapy. And so this idea that the food that we consume really plays a key role in shaping gut microbial ecology turns out to be over a century old now. There's a paper that was published in the Journal of Biological Chemistry that's available open access online if you're interested. It's published in 1999 by Arthur Kendall. And Arthur had done a series of experiments in non-human primates, feeding them different types of food, and then looking at the bacteria in their stool by culturing. And what he described is that differences in the food that he fed the animals changed the types of colonies. And so based on that really sort of simple experiment, he proposed that as food passes through the alimentary canal or the gut, at different levels, it's decomposed by various types of bacteria, and that these types of the bacteria that take part in the digestion of our diet will be determined by the nature of our diet. He was a little bit cautious, and so he pointed out that this correlation between diet and what we now call the microbiota and its end products had been largely overlooked. Despite sort of short flurries of activity, for most of the next century, it remained largely understudied due to the fact that we really lacked the methods to characterize these complex microbial communities without the need for culturing. I was really fortunate when I started as a graduate student at WashU in St. Louis in the Gordon Lab to be able to take part in a series of really early studies looking at the microbial communities found in humans as well as other mammals. And the first project I started on involved a field trip. One of the great things about WashU is that the School of Medicine is right on the edge of this enormous city park. And even better, as a poor graduate student, all of the things in the park are free. And so you can go to the zoo as many times as you want. The art museum is free, the history museum, and there's a bunch of outdoor events that you can go to. We went for a little walk to the zoo. And we told them, we want to study gut microbial ecology. Please give us all your poop. And so they sort of gave us free reign over, although we were monitored carefully, the different animal enclosures. This is Ruth Lay, the first author of the study, and now the director of the Max Planck Center for Microbiome Science. And Sabrina, a really intrepid tech in the lab. They're standing around a big pile of elephant dung, sampling it to sequence which microbes are found in it. And so overall, we looked at 60 different mammalian species that really span the three major types of diet, herbivores, carnivores, and omnivores. They were originally all from the St. Louis Zoo. I was at a conference and I met some people from San Diego, so we got samples from San Diego Zoo. And then Ruth had a friend that is an ecologist that goes and studies Africa. And so we got some wild animal samples at all, and they were from many different types of mammals. And so I'm just going to show you one snapshot of the data that this is one method that we used at the time to cluster microbial communities. So each of these little white names is a different sample that was collected from one of the different 60 different species. And then the little diamonds are bacterial species that we identified. And in this sort of network, they're clustered based on how similar the communities are. And so what hopefully you can appreciate is that all the samples from the omnivores in blue cluster together, this big cluster of herbivores, and then the carnivores go together. And this clustering is only driven by which microbes we found in the samples. And so we didn't bias it at all based on what we knew about the animals. And if you're wondering, humans are just right in the middle of the omnivores. So we look very similar in terms of our gut microbiota to many other omnivorous mammals. And so sort of moving forward, we've now looked at this, the role of diet in shaping the microbiome in a number of different contexts. I told you about the study looking across mammalian evolution, so these really long time scales. But we've also looked at really short-term experiments, so taking lab mice that are normally fed a diet that's really rich in fiber and switching them to a high-fat, high-sugar Western diet. And when we alter the diet of those mice, we can see really profound differences in gut microbial ecology within a single day. Later on, we also studied wild mice, and we found that similar to lab mice, their microbiotas are really sensitive to diet. And of course, this is not just true for mice, it's also true for us. And so we and others have done diet intervention experiments in humans and found that we can see differences in the gut microbiota within days of shifting the diet that we feed to people. And so a lot of the ongoing work in the field really looks at what the consequences of these diet-induced changes in the gut microbiota might be for the host. And one of the things I've always been fascinated with is this concept of energy harvest from the diet. And so just to give you a little bit of context, where you're probably all familiar with these calorie labels or nutrition fact labels that are found in our food. The calories are now nice, big, and bold. You also get information about fat and cholesterol and sodium and other things nutritionists think are important. But what you probably don't realize is that this number for calories is really a biochemical concept that doesn't really take into account much of the digestive process. And so calories are defined as the energy needed to raise the temperature of a kilogram of water by 1 degree Celsius. And the way that this energy is measured is by using something called a bomb calorimeter. It's a tool that we use frequently in our lab now to measure calories in stool or in food. And it's basically a vessel where you put some food inside, fill it with oxygen, and then ignite the oxygen. And then that, as the sample combusts, it sits in this tub filled with water, and you have a really accurate thermometer that can measure how much the temperature in the water changes. And so based on that, people have come up with different conversions for the amount of energy that's thought to be in a given type of food we eat, like cereal or bread. But we wonder, we reason that, a lot of this sort of neglects to account for how the microbiome might affect the food after we consume and the key role that they play in the digestive process. And so we wanted to test if differences in the microbes found in our gut could impact the energy we gain from consuming the same diet. And so this is an experiment that's still impossible to do in humans. And so we turned to our favorite mouse models. And here we took mice that were obese, either for genetic reasons. There's mice that lack the hormone leptin and become chronically obese. And we also looked at mice that were obese due to the diet they were consuming, so eating a high-fat, high-sugar diet. And then we had lean control mice. And then what we can do is take mice that are raised in a sterile facility where they are devoid of any microbiota and introduce these two different types of communities from either obese or lean donors. And we were really surprised and excited to see that the mice that received the samples from the obese donor gained twice as much body fat as the mice that got the sample from the lean donors. And so this really showed that these differences in the microbes that are found in the gut can impact body fat, even though the recipient mice are eating exactly the same foods. And so, this study has, really led to a lot of the ongoing work in our lab and other labs. But I think the most exciting thing for me was that we got a Kathy cartoon. And I'll just walk you through part of it. And so Kathy's friend says the fat gene makes me overweight. Kathy says, no, I tried that one, only worked for a while. Her friend says the stress hormone makes me overindulge. Ho-hum, been there, believe that. The overly efficient intestinal microbes in my system make me gain while others lose. And they both agree we found one we can really stick with this time. And so, we were excited that we had the validation of Kathy. I think one of the major limitations is that a lot of this work was based on mice, and we still lack a lot of really quantitative data looking at the effect of the human microbiome on dietary energy harvest. And so much more, many years later, we collaborated with a clinical nutrition lab, and we're able to show in actual humans that if you perturb the microbiota using a broad spectrum antibiotic, that alters the efficiency of extracting energy from the diet. And so what's true in mice turns out to translate to humans. But that's just one of the many different consequences of changing the structure of the gut microbiota. This is a really nice diagram from June Round that was published last year. And it really highlights the fact that the microbiota can have effects in many different sites in the body. So we were talking about effects on the endocrine system or obesity, but we also now know that microbes can affect the nervous system, skin, cardiovascular system, and others. But I want to sort of caution that, despite all this information, it's still really difficult to predict risk of disease based only on the microbiome. And a lot of that has to do with the fact that we don't fully understand the mechanisms through which these microbes are affecting our body. Another key limitation is that a lot of what we know comes from these gnotobiotic mouse models. And so this is what I referred to in those obesity experiments. And this is just a view inside one of the isolators found in our facility, UCSF. It's an incredibly powerful system where you start with a germ-free or sterile mouse and can really study specific microbes or sets of microbes under controlled conditions. But obviously, these are very different from real humans. And so a lot of the ongoing work in this area is really aimed at trying to make these models a little bit more like humans by humanizing the types of microbes that we put in, or aspects of the mouse's biology, like putting in human immune cells, human cancer cell lines, and others. And ultimately, we really want to understand which of the findings from these model organisms translate to humans and what we can do about it. And so I guess going back about 10 or 12 years, our lab, had sort of realized that a lot of the work in the microbiome space was really focused on risk of disease. And so this sort of thinks about the microbiome very similar to how we think about bacteria or viruses that cause disease. Whereas relatively less was being done to understand what happens after a patient is diagnosed with the disease. And so I got really interested in sort of shifting gears and focusing on how the microbiome might affect response to either dietary interventions, surgeries, or drugs. And just to summarize a few of the different areas that we've worked on, given my background in studying obesity, the obvious question was, what does the microbiome do in response to common interventions for people that are overweight or obese? And so we've done a lot of work on ketogenic diets and are continuing to work on these really interesting diets that are very low in carbohydrate and high in fat. We've also worked on raw food, studying how the cooking process affects the microbiota. Working with the Kaplan lab at MGH, we've worked on gastric bypass surgery. And more recently, we've published data looking at these very low-calorie liquid diets that are used for weight loss. And in all of these cases, these dietary interventions really markedly change the microbes that are found in the gut. And they have really interesting downstream effects for the host. But I really want to emphasize that it's not just about what you eat. It also matters how you prepare the food. So as I mentioned, we've worked on the cooking process. So, even if you eat the same exact food products, the way in which the food is prepared can alter its chemical composition. And so, everybody knows that, potatoes change a lot, whether or not they're fried or mashed and we think that that's important not just for our own body, but also for the microbes in the gut. We've also gotten really interested in food coloring. So, as you all know, the food that we eat is really bright, which is something you don't normally see in nature. And these colorants are really, have many sort of unintended consequences in our gut, and can also be metabolized by the bacteria that are found in our gut in ways that affect how they alter our own body. But I also wanted to just briefly mention that the microbiome field as a whole is also quite interested in this concept of probiotics, or consuming live organisms. And so probiotics are defined by live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. And they date all the way back to Eli Metchnikov, if not earlier, who really popularized this idea. He was actually an immunologist that was really sort of disgusted, I guess, by the colon. He called it a vestigial cesspool in a reservoir for waste matter, in which microbes produce fermentations that are harmful to the host. And so he was obsessed with this idea of somehow fixing the colon. And he learned about these soured milk or yogurt consumed in Bulgaria, and so popularized the use of lactobacillus. But I really want to make you aware that these live microbes that are found in products like yogurt and other probiotics typically don't colonize the gut. This is just one example study published in 2011, where they looked at a popular product in humans. And you can see this particular strain that's in the yogurt is not there in the baseline samples. They then looked at 7 weeks of consumption where it was detected. But then looking 2 weeks and then 4 weeks after the products were taken away, it washes out of the system below the limit of detection, which is in this gray line. And that tends to be true across many studies. These are sort of transient members of the gut, and they tend to be quite different from what's normally found in our body. But despite the fact that these live microorganisms that we consume, either through products like probiotics or through fermented foods, despite the fact that they're transient, they can have permanent effects on the microbiome. This is sort of one set of studies that has really fascinated a lot of us in the microbiome field. It started from studying how bacteria found in the ocean digest seaweed. So they were looking at specific enzymes that these microbes use, and found that they've actually been transferred into bacteria that are found in the gut. And so this is a little bit detailed, but each of these little shapes is a gene. And the blue ones are found in marine bacteria, as well as this gut bacterium within Bacteroides. And so this is sort of something that bacteria can do all the time. They can transfer genes between very distantly related organisms. And this now allows this gut bacterium to digest seaweed. So Justin Sonnenberg's lab, another former member of the Gordon lab, has used this in an interesting way to try to tune the levels of bacteria using these by amending the diet with seaweed. And so here they're looking at Bacteroides ovatus. They put this Bacteroides that can digest these seaweeds into mice. And these mice were either contained a mouse gut microbiota in this RF group, or 2 groups colonized with 2 different human samples. And then they fed the mice either inulin, which is a fiber that the gut microbiota can normally digest, or seaweed, which contains this porphyrin that normally the human microbiota can't digest, but these special bacteria that have gotten genes from marine organisms can. So in the inulin group, you can see that as they sort of switch the mice over to inulin, the Ovata strain was able to colonize the mice that had a mouse gut microbiota, in one of the humanized groups, but it washed through the other group. And then in the seaweed group, you can see that now this circled group actually colonizes much better. And so even though normally it would not be able to colonize, now Ovatus can sort of enter and compete against the other microbes found in the gut. And this technology is now being pursued at Novome, which is a startup company in the Bay Area. And so, all of that is to say that these dietary interventions that are used in medicine can have really important effects on the gut microbiota. But we think that the microbiota is not only relevant for diet, but also really relevant for the drugs that we take. And so in our lab, we study this along the lines of sort of traditional, as a pharmacologist would look at things like a human gene. I think about ADME, which is sort of 4 components of how drugs are handled in the body. They need to get absorbed, distributed in different tissues, metabolized, and then ultimately cleared or excreted from the body. And by far and large, most of what we know about how the microbiome affects ADME is focused on the M, which is why it's a capital. But there is emerging data suggesting that the microbiome affects all 4 components. In terms of drug metabolism, due to a series of sort of focus studies combined with high throughput screens, we now know that hundreds of different drugs can be metabolized by gut bacteria. Many of these drugs are not antibiotics. They're drugs that are intended to target the host or human cells, but they can still be modified by bacteria. And I'm just showing you some examples of our favorite drugs. I think what's really a couple of things that are remarkable is that these drugs span many different disease areas. And so just like, studying the liver can be relevant for many different types of drugs, we think understanding the gut microbiome can also have these broader impacts. And also the structures of these compounds are quite distinct. And so that sort of emphasizes the diversity of enzymes that are found in our gut microbes. In blue, these are all drugs that we are either currently working on or have worked on in the past. And I'll just sort of give you some highlights of a few of these drugs of interest. So for levodopa, that's really an important drug for the treatment of Parkinson's disease. And in a paper published a couple of years ago, working with Emily Balskis's lab in the Harvard Department of Chemistry, we were able to show that 2 bacteria metabolize L-dopa in the gut. The first is a bacterium called Enterococcus fecalis, and it has a tyrosine decarboxylase enzyme that can act on L-dopa, converting it to the neurotransmitter dopamine within the gut. And while that sounds like it might be a good thing, dopamine is actually not able to cross the blood-brain barrier. And so this activation of the drug in the wrong site means that it could lead to less drug being in the brain where you want it to go. On top of that, after this conversion to dopamine, Agarthalolenta, one of our favorite gut bacteria, steps in, and we identified this enzyme DADH that converts dopamine to M-tyramine. Excitingly, we found that carbidopa, which is a drug that's used to block L-dopa metabolism by human cells, does not work against gut bacteria. Whereas in contrast, this related compound, AFMT, is specific to bacteria. And so now we have 2 drugs where we can turn off or on both the host and bacterial metabolism of these drugs. And we're now looking at this in follow-up studies in mice, as well as a clinical intervention study that is ongoing that will look at how perturbation of the gut microbiota with an antibiotic affects drug response. In addition to that, we've also found other ways that we might improve the use of drugs in patients. So in the context of heart disease, we've found specific dietary supplements that can prevent the bacterial inactivation of these drugs, elevating the concentration of the drug that's available for the heart. In the area of rheumatoid arthritis, we've recently published multiple studies looking at how profiling the microbiome can allow us to predict response to treatment. And also looking at how changes in the microbiome in response to methotrexate, a drug commonly used for rheumatoid arthritis, might actually contribute to drug response. And so we think for that particular drug that the off-target effects on the gut microbiome might actually be a good thing, in that they improve the anti-inflammatory effects of the drug. We're also very fascinated about cancer, and I'm going to tell you in great detail about some of the work we're doing there. But we've also identified strategies for lessening the side effects of these drugs, small molecules used for cancer, and also potentially improving the sensitivity of cancer cells to drugs. And so in the long term, we really would, our dream is to have a more microbiome-based version of precision medicine. And so, you know, as many of you have probably experienced, a lot of medicine is sort of trial and error. And so you start with some set of patients, assign them to different drugs that the doctor thinks might be helpful. A subset may respond well, and then the others don't. And so then they come back in again and get a new set of medications, and then that sort of repeats until you either run out of therapeutic options or you've successfully treated each person. For many years now, there's been the idea that we can use the human genome to speed up this process. And so the idea is to sequence the human genome or profile its expression or other methods to try to predict which drug you should use for a given patient and hopefully get to the finish line faster. And what we're proposing is really just a modification of this, where we think information about the microbiome might allow us to more rapidly and more accurately identify the right drug for the right patient at the right time. And so with that, I really want to give you a sense of, you know, the sort of detailed studies that we like to do in the lab. Here I really want to focus on 5-fluorouracil, which is called the fluoropyrimidine. So these are drugs that are really still very important for cancer therapy, and we think have important interactions with the gut microbiome. And so the data I'm going to show you is really started with work done by Peter Spanagopoulos, a former postdoc in our lab that's now in industry. Peter and I were really fascinated by the fact that, you know, although for many years chemotherapy for cancer has been given through continuous infusion, so in circulation, there are multiple oral versions of cancer chemotherapy have been developed. And the reason why we are interested in this is that we study the gut microbiome. And so if you take a drug as a pill, that potentially allows bacteria to encounter the drug before it reaches whatever the target is in the body. And so one example of this is a drug called capecitabine. It's a frustrating drug in that it's highly variable person to person. And despite a lot of research into this, both diet and genetics do not really explain these variations in drug levels in humans. There's also a lot of side effects of this drug. So about a third of patients require some adjustment of their dose due to side effects, and 10% actually have to be taken off the drug. And a lot of these side effects are in the gut, where the microbiome we study is. And there's also some really interesting regional differences in tolerability. So if you take the drug in America, on average, you have much more severe side effects than in places like Europe. And no one really knows what's driving that. In addition to that, we had a number of reasons why we thought capecitabine and its downstream metabolite 5-fluorouracil might interact with microbes. And so, you know, this drug, capecitabine is a prodrug. It gets converted to this compound that's similar to uracil, and that really disrupts central pathways for metabolism that cancer cells need to survive. But the reason why there's a lot of side effects is that those pathways are also important for other cells in our body. And in fact, those pathways are also conserved in bacteria. And so we reason that maybe bacteria would also be sensitive to 5-fluorouracil. In addition to that, our liver uses an enzyme called dihydropyrimidine dehydrogenase to clear this drug from our body. People that have mutations in that enzyme can have really deadly side effects from the use of 5-fluorouracil. Interestingly, there is there had been a work on how uracil, the sort of normal version, the normal compound that's in the body that's similar to 5-FU, is metabolized by bacteria. And we knew that bacteria had conserved enzymes that could reduce uracil. And so we wondered if gut bacteria could actually intercept the drug and lower its levels in the gut. There had also been a series of studies in bacterial cultures, rats and humans, suggesting that drugs like 5-FU alter the gut microbiota, and some really nice data in worms suggesting that if you feed worms different types of E. coli, that can alter how they respond to 5-FU. But what we really lacked is a detailed understanding of how the bacteria that are found in the human gut might interact with these drugs. And so to start, Peter S. Screened a panel of representative human gut bacteria for their sensitivity to the 5-FU as well as other fluoropyrimidines. This is sort of a standard approach microbiologists use to study drug sensitivity. So here we have increasing concentrations of 5-FU, and then on the y-axis you can see the growth of these strains. So for example, this blue bacteria can handle very high concentrations of 5-FU, whereas the purple bacteria is sensitive at these lower concentrations. If we look at that data across all of the strains we tested, you can see that their sensitivity varies a lot. So these circles in red boxes are bacteria that never showed any effect. At the highest concentrations we tested, there was no defect in growth. Whereas in these white boxes, these are bacteria that are very sensitive to low levels of the drug. We went on to study the reasons for this drug sensitivity using the model bacteria E. coli. And through a series of experiments using different media supplementations and bacterial genetics, we found that this really supports the expected target. So, just like in cancer cells, we think these drugs are acting on thymidylate synthase, a really important enzyme for cell growth. But we've also have preliminary evidence that the other pathways might be affected, and so there could be it could be a little bit more complicated in bacteria. But what I really want to focus on for today is how bacteria can metabolize these drugs. And so, as I mentioned, we've shown that fluoropyrimidines inhibit bacterial growth, but what can bacteria do to alter the drug? And so to look at this, we took the top 50% of strains that were most tolerant to the drug, so that the highest resistance, and then screened them for drug inactivation using a really simple disc assay. And so here, what this data represents is a lawn of a very sensitive bacteria, really wimpy bacteria that dies when 5-FU is there. And then each of these discs represents the media, either from controls or after growing bacteria. And so what happens if the drug is there, there's this zone or circle of clearance around the disc, indicating that the drug there has killed off the indicator strain. And so this is just a control to show you that the drug is stable over the 3-day incubation with media. We then screened it across our panel of strains, and you can see that the zone is there in almost all of these. So, most of this looks identical to the control, with 2 exceptions. And so here you can see coli, the zone goes away by that 2-day time point. And the same happens for Salmonella enterica. So 2 of the strains that we tested inactivate the drug. And we went on to use mass spectrometry to show that that was due to the reaction that's really identical to what happens in the liver. So here, the drug 5-FU gets converted to this downstream metabolite dihydrofluorouracil in blue by both of these bacteria. And so because we knew a little bit about how bacteria metabolize uracil, we thought, or we had a good guess for which bacterial genes could be responsible. And so in humans, as I mentioned, there's this enzyme called DPYD. It's expressed by a single gene. Whereas in E. coli, DPYD has been split into 2 different genes, pre-T and pre-A, that are thought to carry out the same function. And so we wanted to ask the question of whether or not pre-TA is necessary for inactivating 5FU. And so to do this, we worked with the model bacteria, gut bacteria E. coli. And then we used our DISC assay as well as mass spec to monitor metabolism in different strains of E. coli. And so these are just controls. This is the media and then an empty vector that we use to express the gene. You can see again, at the end of a 2-day incubation, it's all 5FU. When we look at wild type E. coli, now after 24 hours, you can see the zone is decreased and the 5FU has been converted to GHFU. Whereas in the bacteria that don't have pre-TA, they look like the media controls. And we also showed that these genes can rescue the knockout. So in a bacteria where we've deleted those 2 genes, if we put them back on a plasmid, they can now produce DHFU. And so that really shows that these genes are necessary and sufficient for these bacteria to inactivate this cancer drug. More recently, Ben Guthrie, a really talented protein biochemist postdoc in the lab, has been able to purify these 2 proteins from bacteria. So this is just his slightly fancier version of the different domains that are found in these 2 proteins and a predicted crystal structure. If there's any biochemists in the audience, this is just evidence that the cofactors that are important for the enzyme to function are there. And then now that we have purified pre-TA, we can now add either 5FU or uracil and monitor the reaction over time in a test tube. And so both of the predicted things occur. What's really more interesting is that now we can measure the kinetic or the efficiency of the enzyme in these conditions. And so you can see that both 5FU and uracil are used sort of similarly by the enzyme. Whereas when we look at the reverse reaction, so the dihydrouracil going back to uracil, it's much slower. And so just to give you a sense of that, we require about twice as much of the substrate for the enzyme to do anything. And there's also a lot less formed here on the y-axis. And so we think what this means is that this reaction is essentially irreversible, that the bacteria are converting uracil or 5FU in a single direction to these inactive reduced forms. And so that potentially suggests that the gut microbiome may effectively clear the drug from the body. And so that would lower the amount of drug that's left over to treat the cancer. And so we wanted to turn now from these studies in the test tube to studies in a mouse model that actually has cancer. And so to do that, Than Kya, a really talented graduate student in the lab, worked together with Joyce Lee, a postdoc in Andre Goga's lab, card-carrying cancer biologists. And there's a lot of sort of pieces to this model to walk you guys through. So we're using human cancer cells in mice. And so because in a normal mouse that would cause an immune response, we have to use what are called nude mice that lack a mouse immune system. We can then put these cancer cells in as a xenograft, so injecting it into the skin. We then use an antibiotic, streptomycin, to clear the gut microbiota, and then allows us to colonize these mice with different strains of E. coli. And so, for example, we can put in the E. coli that lacks free TA or the E. coli that expresses free TA. And then, once we've established that model, we can sort of put them through a randomized control trial like you would do in humans, where we randomize these mice to either control or getting the drug capecitabine. And so just, this is, our sort of expected result before doing this study is that in the mice colonized with the knockout strain, the drug would work well to decrease the growth of the tumor. Whereas in the mice colonized by the pre-T expressing strain, that would lower drug levels and reduce the efficacy of the drug. And so just as a control, we looked at the level of the tumors at baseline, and we found that they were very similar across all the groups. But what was really exciting is that we found exactly what we predicted in terms of the effect of pre-TA on drug efficacy. And so here you can see these sort of pink open circles are tumors that are really halted in growth by treatment with capecitabine, whereas the other three groups all have tumors that continue to grow, including this pre-Ta positive colonized group that's getting the drug. And so that suggests that these bacteria are intercepting the drug before it reaches the tumor. This is just another way to look at the data. So 17 days after treatment, we see that there's smaller tumors in the knockout group, but not in the pre-Ta positive group. And even more strikingly, at 17 days, we randomly selected 4 mice per group to dissect out the tumors. And you can see that they're much smaller in the knockout group versus the pre-Ta expressing group. And we think the reason for this is that pre-Ta is decreasing the bioavailability, so the ability of this drug to get absorbed and reach the tumor. And so to look at this, we've done a variety of different experiments where we measure drug levels over time. So here we start with completely sterile germ-free mice, colonize them with our different E. coli strains, and then dose them with the drug and measure drug levels over time. These are just a couple of controls to show that the mice are similar weights and that they're colonized at similar levels with these 3 different strains of E. coli. But when we look at drug levels over time, as we expected, the knockout has the highest levels of drugs in circulation, whereas the strain that expresses the highest levels of pre-Ta in green have the lowest levels of drug. And so all of that was, really focused on this model organism E. coli that can affect capecitabine both in vitro or in a test tube and in mice. But we wondered whether or not E. coli was really the most important bacteria to worry about, or whether or not there could be other bacteria in the gut that have the same genes or enzymes. And so working with Patrick Bradley, a former postdoc in Katie Pollard's lab here at the Gladstone at UCSF, we analyzed a previously published set of 575 samples that came from colorectal cancer patients and controls. And what was really remarkable is that we were able to find these genes in every single person that we looked at in the study. So all 179 were pre-Ta positive. And there were actually only 6 samples. So, the study had multiple time points per person. And so there were 6 samples that were negative for the genes. So this really suggested that these genes are always found in the gut microbiome. But what was really different is, what varied person to person, is that the levels or abundance of these genes in the gut was quite variable. And so that's just shown here. You can see this is on a log scale. So there's orders of magnitude difference in the abundance of these genes in the gut microbiome in both healthy controls as well as cancer patients. We could then use that data to ask which bacteria harbor pre-Ta in the human gut microbiome. As expected, we found pre-Ta in E. coli. However, that was just a subset of the people in the study. The vast majority of the people actually had pre-Ta from a very poorly studied group of gut bacteria called anaerostypes. And so this has now really caused us to focus our attention on the anaerostypes and really think more deeply about what role they may play in all this. We've also started to look at how these genes and others may be altered during the course of therapy. And this is work done in collaboration with Wes Kidder in the UCSF GI Oncology group, as well as Chloe Atreya and Alan Vanook and others. One of the studies we're looking at is called GO. It's the gut microbiome and oral fluoropyrimidine study in patients with colorectal cancer. And we're nearing the end now of looking at 60 different patients that were all taking either capecitabine or other types of fluoropyrimidines orally. And so one of the interim analyses that we did was just looking at the first set of patient samples that we had over the course of therapy. And 2 of them were only given capecitabine, and the rest of the patients were given a combination of capecitabine and immunotherapy. And what we're graphing here is just the abundance of pre-Ta, so that one specific set of bacterial genes, but you could do this for any other gene of interest in the data. But what I really want to draw your eye to is that in patient 13, you can see that pre-Ta is really stable over time, Whereas in patient 1, there's this massive expansion or increase in the abundance of pre-TA. And we're really curious whether or not these differences patient to patient and how these gene levels are altered correlates to side effects or the efficacy of treatment. And consistent with the previous analysis, by far and large, the anaerotypes types are the major contributor to these genes, but E. coli also was important for a subset of patients. And so the source can vary person to person. And so just to summarize this data that I showed you, hopefully that wasn't too much detail, but I wanted to give you a sense of a sort of recent and ongoing study in the lab. We've shown that gut bacteria can inactivate 5-FU in an essentially irreversible manner. This inactivation decreases drug efficacy and drug levels in mice. And we've also found that human cancer patients vary in the abundance of these pre-TA positive strains. And one thing that this is really highlighted for us is that it's really hard to attribute any metabolite that you can measure in the body to host versus microbial enzymes. So as I mentioned, this reaction that the microbiota is participating in is identical to what the liver does. And so if you measure dihydrofluorouracil, there's no way of knowing whether or not it came from the microbiome or from the liver. And so we're really hoping to address that, the relative contributions of the host versus the microbiome in future studies. But, as is probably clear, we've got a lot of work to do. Really, any of those sort of basic questions that a journalist would ask related to the role of the microbiome in pharmacology are still open. This includes who are the most important bacteria to study, what drugs are affected, how exactly this works. So, what are the details in terms of the enzymes or other mechanisms through which bacteria affect drugs? Where is this occurring? When is it occurring? So both in terms of short timescales, right after a drug is taken, within hours, or within weeks or months, as patients are taking drugs over time. And perhaps most interestingly, why does any of this happen? So, why did the microbiome evolve? Enzymes that act on drugs, and what is in it for them. In my remaining time, I wanted to just point out that the gut may not be the only site in which bacteria can affect drugs. This is an interesting study that was done by another lab looking at the interaction between bacteria inside the tumor and drugs. And so here they focused on gemcitabine, which is a drug used for pancreatic cancer. And what they found is that Enterobacteriaceae, which include E. coli, can actually invade the tumor itself. And there they identified an enzyme through which E. coli can inactivate gemcitabine. And also in mice showed that if you clear these bacteria using antibiotics, you can increase the sensitivity of these tumor cells to the drug. And so I think, this, as well as many other studies looking at the tumor microbiome or the microbes found within tumors, really highlights that there can also be local effects of bacteria on drugs acting at the site of the tumor. And these effects also extend beyond small molecule chemotherapies. And so for those of you that aren't aware, there's been this revolution in immunotherapies for cancer. We're really fortunate to have Max Krummel at UCSF, who was working in Jim Allison's lab at the time when he studied negative immune regulation that eventually led to the Nobel Prize in 2018. And just as sort of a high level, microbiologist view of how this works, tumor cells are targeted by immune cells, and these immune cells express different receptors that can have positive effects that sort of drive their ability to clear the tumor cell. And then what's called a checkpoint, or these negative inhibitory receptors that sort of tamp down on the immune cell's activity. And so there's been a lot of interest in whether or not the microbiome could affect response to these drugs. The drugs that target these inhibitory receptors. So here, this is an antibody that targets the PD-1 receptor that sort of shuts down these immune cells. And so just, conceptually, it's important to think about why gut bacteria could affect immunotherapies. So this is a commentary from Lauren Zutbogel, an active scientist in this area. She talks about how these drugs affect the barrier function of the gut. We know that the microbiome has a lot of important interactions with the epithelium, with the immune cells that are in the gut, as well as other tissues, as I mentioned earlier. And also the microbiota has a systemic effect. So it can actually affect the secretion of cytokines, and the microbes themselves produce metabolites that can control immune function even outside the gut. And so one of the really influential studies in this area was done by the Gajewski lab at University of Chicago. There they focused at, or they found this really interesting difference between mice that were from two different vendors. And so mice from Taconic, one company that sells mice, versus Jackson Labs, had really different trajectories for tumor growth over time. And so here you can see in the untreated mice in blue versus red, the Jax mice actually have slower growing tumors. And that ends up in sort of improving their response to checkpoint blockade, these immunotherapies. They then showed that you could colonize Taconic mice with the microbes from a Jackson mouse, and that would improve response to immunotherapy. And this is just some sequencing data identifying different groups of microbes that are found in these Jax colonized Taconic mice. And so at the top, these are candidate tumor inhibiting bacteria, and at the bottom, candidate tumor promoting bacteria. And they went on to show that one specific group of bacteria that were potentially tumor inhibiting, the bifidobacteria, improved response to checkpoint blockade. There's been a series of other studies, both in mice and in humans, following up on this. Here's a study from 2018, again showing that in human melanoma patients, that the people that respond best to checkpoint blockade have higher levels of bifidobacteria. But I'll note they also have higher levels of these Corynebacteriaceae that our lab is really fascinated by. So maybe it's not all about the bifidobacteria. And so, given this interaction between the microbiome and immunotherapy, as well as chemotherapies, there's been a lot of interest in how we might use this to improve cancer therapy. And I just want to touch on one potential application of fecal microbiome transplantation. And so that, FMT has been, by many sort of touted as a potential miracle that could be used for many different disease areas. But I think it's important to note that this is not a new concept. And so FMT, or fecal microbiota transplantation, actually dates all the way back to the 4th century in China, where Gi Hong, a really well-known Chinese medical doctor, used human fecal suspensions by mouth for various diseases. The interest in this intervention continued, and in the 16th century, there was a lot of reports about another individual that really tested a lot of different preparations of feces for medicine. And they affectionately called it yellow soup. More recently, Alex Kurutz and many other gastroenterologists, including people at UCSF, have continued this approach, both for infections like C. difficile, as well as other indications. But I really want to point out that the general approach is really not that different from what was done back in the 4th century. Last year, there was a lot of excitement around 2 studies that were published in the same issue of Science, showing that FMT may actually improve immunotherapy responsiveness in human patients. These are small pilot studies, one done in Israel, where they took 10 people given these immunotherapies and gave them all FMTs from 2 different donors. And you can see 3 of the people actually showed pretty remarkable reductions in their tumor size over time. The second study was in Pittsburgh. They had 15 individuals, and depending on how you sort of slice the data, somewhere between 3 to 6 of those 15 people showed a good reduction in tumor size in response to this combination FMT and immunotherapy. But FMT is not perfect. There is a very important description in 2019 in the New England Journal about a bacteremia, or bacterial infection of the blood, that occurred in 2 different patients that were given an FMT that carried a drug-resistant E. coli. And there's also potentially other consequences that we don't know about due to the many ways in which microbes in the gut interact with the host. And so this has really driven a renewed interest in replacing FMT with other alternative approaches. And so I'll just sort of at a high level describe a few of these ideas. So one involves really taking a traditional pharmacological approach to targeting the microbiome. So, once we identify specific enzymes or bacterial targets, we could potentially develop drugs that inhibit them. And a proof of concept of this relates to cancer therapy. So a drug, irinotecan, has a lot of side effects due to its bacterial metabolism in the gut. And Matt Redinba's group has been able to identify drugs that inhibit this bacterial enzyme. And in mice, that can really decrease the GI side effects of irinotecan. There's also a lot of interest in depleting the specific members of the community. So this is sort of a more targeted version of the antibiotics that we currently have available to us. And that could be done with drugs or diet, as well as through the use of viruses or bacteria, phage, that target these bacteria. And there's also a lot of excitement around engineered bacteria. So, trying to introduce new live organisms. This is sort of similar to the classical probiotic approach, but maybe takes advantage of our ability to now manipulate the genomes of these bacteria. And so, for example, there are engineered bacteria, and people have made bacteria that can rapidly metabolize the amino acid phenylalanine. And these are able to decrease the overall levels of this amino acid in the blood of mice, sort of mimicking a human disease called phenylketonuria. And then finally, we and others are really excited about this idea of actually modifying the genes of bacteria within an animal. And so, one way that this has been done by Harris Wang at Columbia has been through introducing E. coli that can transfer genetic materials through a process called conjugation into other bacteria that are in the gut. We've also been really interested in this idea of harnessing both these viruses that infect bacteria or phage, as well as these the remarkable innovations around CRISPR systems that allow you to target specific genes. And so really the dream is that we could avoid these off-target effects of drugs on many different members of the community and really precisely target one member, either to remove it or to cause it to have a new function. And as a proof of concept, we've recently published a system where we have done this in mice that are colonized with E. coli that we can target with a virus called M13. And we can use that to remove specific strains from the community or even specific genes. And so with that, I want to thank you all for your attention. I know that we covered a lot in the last hour. I want to thank the lab and everyone that contributed to this and our funding sources. Also, as a, I guess, shameless plug, I have a podcast called Science is Funcast, so feel free to check us out or visit our lab website or the Microbiome Center to learn more. And so with that, I'm looking forward to the questions, and I'm happy to hear what you guys think.
That was really great. Thank you so much, Peter. A tour de force. So we've got a number of questions from the audience. We'll kick off with the first one. It's a question about how stable or permanent are these diet-induced changes? And so this is someone who's trying to reconcile the notion that, one's kind of core microbiome may be established in early life and childhood versus the literature showing that the changes in the gut microbiome by changing the diet. Mm-hmm.
Yeah, that's a great question. So, I told you one example. Is that I'm an awesome nerd? Yeah, I showed you like one example of a potentially permanent change, where these marine organisms have transferred genes into bacteria in the human gut. And so that we think happened a long time ago and is sort of stuck around in the human microbiome and is now propagated between bacteria and humans. That's maybe a very extreme and specific example. So, in contrast, in the sort of intervention studies that we've done in mice and humans, we typically see that the microbiome goes back to its baseline state really rapidly after we return them to the original diet. And so that's sort of good news if you think about, over the weekend, if you sort of, go out to eat and you eat a lot of junk food, we don't think that those changes will be permanent. Then, when you go back to your normal diet on Monday, your microbiome can recover.
Great. Yeah, that addresses one of the other questions that another attendee had. But the next one is, why do the probiotics not stick? And maybe prebiotics might work better. Your thoughts on that?
Yeah, that's a great point. Part of what is, I guess, now happening is a new generation of probiotics that are really, built off of the knowledge we now have about microbial ecology. And so selecting strains of bacteria, like the Ovatus example I showed for porphyrin, where, these are bacteria that are really widespread in the human gut and are maybe better at competing and gaining some traction. And that's really different from more traditional probiotics where, those were selected because they're easy to grow and, really tolerant to things like oxygen and can be, prepped in really large scale. And so they weren't really selected for stable colonization. On the flip side, I think there is an argument to be made that there's a lot of value in these transient colonizers because, a lot of what the worry around engineered bacteria or these more next gen probiotic products is that, is around biocontainment. So, we don't want to release, novel organisms into the world. And so, we may actually want to engineer them to be relatively transient.
Yeah, it's a really good point. There's another question. Is there parallel research on the skin microbiome, the local and systemic disease, and the effects on topical medications, including 5-FU and those niches?
Yeah. I don't know if the person asking is a dermatologist, but I've been trying to get somebody to work on that. I think the sort of gut-skin interaction is interesting for a number of reasons, both for what you mentioned, like, in the gut, we have to think about how drugs, that are taken orally, there's sort of a race between the ability for the microbes to encounter the drug versus the drug getting absorbed and leaving the gut. Whereas for topical medications, you have a lot of direct contact of drug, at the site where the microbes are growing. And so I think that's a really interesting question of whether or not, the same drug could have a stronger effect as a topical medication. In addition to that, there's an interesting idea of sort of transit of drugs, from the skin to the gut or from the gut to the skin. And so, that's a sort of emerging area of research looking at, how antibiotics that are taken orally might actually affect microbes in the skin or other sites in the body. I'm also curious about the other direction. If you put a drug topically, does any of that return to the gut?
Lots of areas for lots more research for many more years. So the next question relates to food colorings, and someone who's heard that red in particular is quite noxious. And the question is, do all food colorings behave the same?
Yeah. So I don't know if you were thinking of red number 40, which is our favorite. I don't know if or maybe least favorite food coloring, but there's a lot of different compounds that are used as colorants, and we think they are definitely different. So the red number 40, as well as other commonly used food colorings, have a sort of special bond that we call an azo bond. So it's just two nitrogens that have a double bond in between them, and that's for people that study bacteria is sort of famous because our own body can't really handle those bonds, whereas bacteria are really good at breaking them. And so that was actually the reason why our lab originally got interested in food colorings, because we found that a subset of food colorings that contain the azo bonds are broken down by bacteria. But there's other reds that don't have an azo bond, and so we think that maybe that would be safe from bacterial metabolism. So yeah, I mean they definitely vary a lot in structure and could all have very different effects on the microbiome.
Great. I know you addressed this. You said it was a quick return, but there's a question: how long does it take the composition of the gut microbiome changes to return after, for example, a healthy or an unhealthy diet? Maybe more specifics in how long it'll take before after you've trashed your microbiome, how long before it comes back to normal again?
Yeah. Yeah, I mean, I think there's relatively, we need to do more work to sort of iron that out for, there's so many different diets that people ingest. I guess one study that we did was a very extreme dietary perturbation. So we fed people, we either had them only eat plants for 5 days or only eat meat for 5 days. So this was sort of inspired by the zoo study I told you about, where we wanted to make people herbivores or carnivores. And on the all meat diet, we saw a really remarkable effect on the community. And that we could detect within really the first day after they were on the diet. And then after they were released from eating all meat, we saw the recovery happen just as fast. So, within a day or two, things sort of got back to normal. I think one of the questions moving forward that we and others have is, what happens if you sort of stay on a diet, maybe not as an extreme diet as that, but, if you're on, if you're eating a high meat diet or, a high vegetable diet or whatever for long periods of time, is there sort of a tipping point where, your microbiome can no longer go back? And there is some data from around Ilanov and others suggesting that there can be permanent effects if you've sort of had been on a diet long enough.
Maintain that selective pressure over long periods of time. Yeah, makes sense. Next question: in colon cancer, and this question came in when you're talking about drug metabolism. So in colon cancer, how might the surgical removal of a part of the colon affect the process of drug metabolism and the drugs that are administered afterwards?
Yeah, that's a really interesting question. We're hoping to get at that in some ongoing studies with our collaborators in the Cancer Center. There's a lot of interest in sort of the timing of chemotherapy versus surgery. So, whether or not, that's really more focused on, removing the primary tumor. But I think in terms of like taking out the whole colon or the large part of the colon, I'm not sure exactly what would happen. Maybe Sue knows more than I would.
Pancolectomy, you will lose all of the distal gastrointestinal tract, and typically there's a subsequent surgery to generate a J pouch, which is made from the ileum, which is a kind of a pseudo colon, but it's not a colon. It doesn't have the same microbial content, and so you might imagine that the metabolic capacity of that microbiome and the J pouch is fairly different from what you'd have in a colonic microbiome. Mm-hmm. So yeah, but we have the samples if you want to check it out, Peter.
Yeah, that's an interesting question. Yeah, and I've heard, I guess, like in talks, like in the absence of the colon, that the sort of distal small intestine becomes more like a colon, right? Yeah. So I mean, it would be interesting to know if that like transfer, does that move some of the colonic metabolism into the small intestine?
Yeah, it's quite minor though compared to the activities that you would have along the length of the colon.
Yeah.
So someone would like you to just reiterate what pre-TA is again?
Yeah, so pre-TA is just 2 genes that are found in E. coli, and together they make up an enzyme that is similar to this enzyme DPYD that the liver has. And so it's basically just a protein machine through which bacteria metabolize the drug 5-FU. as well as uracil, which is just an important compound for all cells.
Great. In your early yogurt slide, what is the takeaway from your finding that the study bacteria were not in the original microbiome?
Yeah, I mean, I think that's sort of related to what we were talking about earlier, that, a lot of these strains are not, they weren't even isolated from the human gut. So they're sort of commercial strains that are comfortable in yogurt and other food products, but they are not necessarily widespread in the human gut.
Mm-hmm.
I mean, I will point out that one of the challenges we have more generally in the microbiome is that, which was on that slide, is, it's hard for us to sort of measure things that are at very low levels, given that, in the gut, there's 10 to the 11, so there's hundreds of trillions of cells. And so, if one or two of those cells, is the probiotic strain, it's hard for us to capture that. And so, it's always hard to exclude the possibility that maybe those probiotic strains were there, but they were at really low levels.
Is it thought that many medication side effects are really representing interactions with the gut microbiome? Is that why so many meds cause GI side effects?
Yeah, I think maybe. Yeah, I mean, it's something that no one until recently has really paid much attention to. I think there was, we know that many drugs cause GI side effects, but a lot of times we don't know exactly why that's happening. And it was really only, in the past few years that we've gained an appreciation that many of these drugs matter for bacteria. And a lot of that is, I think, is really surprising. For example, statins, which are, one of the most widely used drugs in the world, for cardiovascular disease prevention, are they target an enzyme that bacteria don't have in the gut, and so they shouldn't matter for bacteria, because the gut bacteria don't actually have the target of the drug, but we and others have shown that they still do really matter for bacterial growth. And so, I mean, that just goes to show that even if we've identified one mechanism through which drugs act, they can still have many other effects on cells that we don't anticipate.
Well, if they change the host, they change the ecosystem, the environment for the microbes, and that have an indirect effect on the microbes that are there.
Yeah, that too.
The next one is a very specific question. What is your estimate? We're going to hold you to this, for the total number of species in the usual human microbiome.
Yeah, I mean, it sort of gets into the weeds of what a species is and how we define that, and a lot of boring sort of technical details. I guess ballpark, I think we're currently hovering in the hundreds of species for a typical person, but that can change pretty dramatically depending on what diet you're on or what drugs you're taking.
And that's in adults as well.
In adults, yeah, exactly.
Next question. I've noticed a few oncologists recommending NAD plus supplementation. Is this related to the microbiome?
Yeah, I don't know. I haven't looked into that, so I'll have to check.
Yeah, not sure either. Next one. How does stomach acid and digestion affect or does not affect application of probiotics?
Yeah, that's a great point. So I guess another reason that I didn't mention before, the strains that have been selected traditionally for probiotics are either chosen to be tolerant to acid, because obviously, this is also true for food and drugs. Anything that we consume has to make it out of the stomach intact, or at least somewhat intact. And so we definitely think acid is important. It turns out, I guess, through studies and mainly in these notobatic mouse models, that's turned out not to be a huge barrier for the normal members of the human gut microbiota. And so that it's maybe not surprising, given that they have to get to the next gut after you have a child and pass on your microbes. And so that, they tend to be relatively tolerant to acid. or at least tolerant enough so that there's still cells remaining to colonize the rest of the gut.
Right. Next one, has a ketogenic enhancing microbiome been identified?
Like microbes that drive ketone body production. Yeah, that's a great question. Yeah, it's actually something we're revisiting now. So one of our collaborators that we're working on together with is a guy named Peter Crawford. He's at the University of Minnesota, and he trained in the Gordon Lab. He was there as a clinical fellow when I was a grad student. And one of Peter's studies, Peter C, looked at germ-free mice versus colonized mice, fed ketogenic diets. And what he found is that at least in those mice, that the germ-free mice didn't produce as much ketone body, and said that there was something sort of defective in their body's ability to go through ketogenesis. And we're currently sort of revisiting that through more controlled experiments where we put in certain types of bacteria, but nobody's really sort of figured out exactly why that is. Is it what is it that the bacteria are doing to the liver or to other organs to affect the production of ketone bodies?
The lungs must have an interesting microbiome as well, and I bet they play a role in immunity, and who knows, maybe the transfer of gases and nutrients. Do you have any thoughts about the lungs and bacteria?
Yeah, I don't have much to say on that, Sue. You do.
Well, if you come to the American Thoracic Society meeting early tomorrow morning, I can give you the whole history. I'm presenting the whole history of the lung microbiome and how it relates to chronic inflammatory airway disease, and how we're leveraging efforts to try and examine the lung microbiome, the airway microbiome, to understand different endotypes of diseases like chronic obstructive pulmonary disease and asthma, and determine novel approaches for treating subsets of patients with very different microbiomes in their lungs that have very different immune phenotypes and clinical outcomes. So in parallel with the gut microbiome, it's a little bit behind, but it's catching up in terms of leveraging that microbiome to understand chronic airway disease.
Yeah, and I think that's the lung is also maybe, like the skin, another opportunity to think about drug exposures where, sort of inhaled medications might, more directly go to the bacteria that are found in the lung.
Yeah, and we also see, corticosteroid using patients, like patients diagnosed with asthma, have a very different microbiome and corticosteroid degrading microbes in their airway microbiome. So yeah, it's an
interesting
area ripe for an analysis.
Mm-hmm.
Peter, an audience member wants to know what alcohol does to the gut microbiome.
Yeah, that's a great question.
Or hot tea. There's a second part.
Or tea. Yeah, I mean, this is sort of related to this, but one of the sort of fun signals we found in one of our diet studies. So we were taking samples every day from people for multiple weeks and sequencing their microbiomes, and then we got this sort of weird. We looked for fungi or small eukaryotes that are in the gut, and we had sort of a distinct signal every Friday night or every Saturday after the Friday. And we were actually picking up Saccharomyces from the beer that the participants were drinking. So there's definitely, I think, in alcohol containing beverages, a lot of interesting stuff that could, we think, feasibly affect the gut microbiome. That includes, microbes that are in fermented beverages, but also in drinks like wine, there's a lot of polyphenols that have a lot of important interactions with gut bacteria. In terms of alcohol itself, I don't know. I mean, there's definitely some interesting studies that other labs have done looking at alcohol-induced liver injury and its relationship to the microbiome. But yeah, I'm not aware of data suggesting that like the ethanol itself sort of changes the bacteria. I mean, obviously we know that ethanol is really toxic to bacteria in a test tube. But when you drink alcohol, then ethanol leaves the gut really rapidly, and so it may not actually have a big effect on directly on the bacteria.
We have quite another number of questions, so I'm going to try and get through as many as possible in the last 6 minutes. But next one is, how have our microbiomes drifted over human evolution? And does eating a Western diet lead to a less healthy microbiome?
Yeah, I mean, that's a hard thing to answer. I think, as you're probably aware of through this course, like most of the data that we've gathered has been over the last, 15 to 20 years. And so, we've gotten a lot of detailed information about how the microbiome has looked, in the 21st century. It's hard to sort of look back in human history to get a sense. There is some really interesting data from fossilized stool samples called coprolites that give us, some history. And then there's also, work sort of looking at different populations of people that live different lifestyles, like hunter-gatherers, to try to sort of approximate, past lifestyles. But yeah, we don't, the answer is we really don't know sort of what the microbiome looked like in the past. In terms of diet, we definitely know that the sort of high-fat diets that we consume in the Western world have a really major impact on the gut microbiota. And there's been a lot of work trying to understand what the consequences of that are. We've looked at that in the context of driving obesity. Other people have looked at inflammatory bowel disease and cancer. So there is, some thought that a lot of those changes may be negative.
Yeah. If the mother is vegetarian, is it more likely that the infants will also prefer that diet? Or if vegetarian is considered healthy, the infants will be healthier and have a healthier core microbiome?
Yeah, that's a great question. I mean, we haven't really done any of this work in our lab, but there's a lot of really, I think, fascinating data suggesting that the diet of the mom really matters a lot for their offspring. And there's a lot of different reasons why that might be true. Development in utero is really important. As you can probably comment more on this than me, but there's also evidence that if you put a pregnant mouse on a high-fat diet, that there can be really prolonged changes in the microbes that are in the gut of her babies. And so, I think that's a really interesting area to look at more in the future.
And intergenerational extinctions over time, just with that first kind of dietary intervention in the mother mice. How might fasting, intermittent or prolonged, impact the microbiome?
Yeah. So that's a great question, and it really gets back to our fascination with these ketogenic diets. So, ketogenic diets are they're basically a way to avoid the need to fast. So, fasting has been used since antiquity to treat or prevent disease. But as anyone knows, if you've ever tried to fast, it's really hard. And so, fasting induces the process of ketogenesis, so the breakdown of fat to form these ketone bodies that feed the different tissues in our body. And it's really similar to the process that our body goes through when we're on these really low carbohydrate ketogenic diets. And so we've shown that in unpublished data that we can recapitulate, or we find similar effects of fasting to ketogenic diets. And interestingly, other labs have looked at intermittent fasting, where even short periods of fasting, like controlling when you eat, can sort of counter some of the effects of a high-fat diet on the gut microbiota.
Great. Have you done any, or do you know any studies about chronic levels of stress hormones like cortisol affecting an individual's native microbiome population, and or the effectiveness of reseeding the microbiome if depleted? And this person is thinking about chronic stress as a contributing factor to diseases like cancer. Yeah,
I haven't really looked too much into stress, although I'm embarrassed to admit that, given that UCSF has a center called SSEW. It's a Sugar, Stress, and Environmental Weight Center. And there's a lot of really fascinating work at the sort of intersection between diet, stress, and obesity and metabolic disease. But yeah, we've sort of in our lab really focused on the diet side of that equation, but maybe because it's easier to model in mice. But
I recall a study with probiotics given during antibiotic therapy actually showing a delay in recovery of our normal gut microbiome, thus potentially interfering with rather than promoting gut recovery. So should we really not be promoting the use of probiotics during or after antibiotics?
Yeah, I think it's a really good point. And I think that's just one study, but we need to do a lot more work to understand what the best solutions are after antibiotics. So, there's been a lot of interest in maybe using FMT to sort of reintroduce microbes after antibiotics. But could we get away with cocktails of microbes that are normally found in the gut, or other sort of more rational interventions? I will say, I always thought it was weird that people recommended probiotics after antibiotics because of the reasons we talked about earlier. These are actually not the normal gut bacteria, and so I would, I guess it makes sense that maybe introducing these sort of foreign organisms to the gut might sort of interfere with the bacteria that we want to recover.
So, second last question, if you'll indulge me. It's about Parkinson's disease, and the thought that it develops in the gut, and if that's so, shouldn't it be possible to treat the disease through interventions on the microbiome?
Yeah. So that's kind of what we're looking at in this clinical study. There's actually already been a couple different studies that use antibiotics in Parkinson's disease. These are both, small studies, but suggesting that antibiotics may improve response. We think one of the reasons why it could be antibiotics could be good is getting rid of those bacteria that intercept L-DOPA. But it's also possible that if there are bacteria that are contributing to the disease itself, that the antibiotics could be acting in other ways. And so we're trying to sort of deconvolute those different effects of antibiotics, both in the human clinical study as well as in mouse models.
Right. And then the last one for tonight, how do PPIs and antacids affect the gut microbiome?
Yeah, it's really interesting. So I don't know about antacids in terms of like the daily antacids that you might take after a meal, but definitely the sort of prescription drugs that are used for gastric reflux and others have been they've been really highly associated with differences in the gut microbiome. And so this has really come out in more descriptive surveys of the human gut microbiome, where people have tracked the drugs that people are on in large populations and found that those drugs do correlate with changes in the structure of the microbiota. So I'm not aware of a lot of like detailed, I think it's still sort of an open question why that matters. Is it sort of affecting the levels of bacteria as they get introduced in the gut, or are there effects of those drugs in the more farther down in the colon? But yeah, it's a really interesting question.
Great. Well, Peter, thank you so much. That was really, really wonderful. And thanks to our audience. Thanks. Have a good evening and take care.